Hello, and welcome back to Father-Offspring Interviews. We have reached episode 30, so let's get this party started. Patrick from New Hampshire asks, what do you think the mechanism behind PSSD, Post SSRI Sexual Dysfunction, could be? Some people report sexual issues and even anhedonia lasting for years after taking SSRIs for only a couple of weeks in some cases. Could it be due to SSRI-induced neuroplasticity?
Okay, lots of acronyms to unpack here, but the general punchline is, as usual, there is no free lunch. Okay, you got two neurons. One of them sends out a long cable reaching out to another one's cable. They don't actually touch. There's a space in between called the synapse. In order for this neuron to talk to that one, it releases chemical messengers, neurotransmitters that go floating across the synapse. And those neurotransmitter chemicals then bind to receptors on this neuron, and this neuron gets all excited. So this neuron has sent this message to this neuron. This neuron only had a brief thing to say. It doesn't want that neurotransmitter to stick around there on the receptor on this side for hours afterward. You got to get rid of it. Neurotransmitter comes floating off the receptor, and while it is in the synapse here, the neuron has two options. It could break apart the neurotransmitter and flush it down the toilet, your urine, your cerebral spinal fluid, or there could be a pump at the end of this neuron that will take the neurotransmitter back up to recycle it, to reuse it next time, a reuptake pump. So SSRIs, leading drugs for depression. SSRIs, best known Prozac, fluoxetine, also Paxil, selective serotonin reuptake inhibitors. Serotonin is one of those neurotransmitters, and there's a pump that takes serotonin back up into the end of this neuron and recycles it. And thus, if you take a selective serotonin reuptake inhibitor, you block the reuptake. The serotonin sticks around in the synapse longer, and for lack of anything else useful to do, it hits the receptors here a second time and a third time and a 50th time. So throwing in a reuptake blocker amplifies the neurotransmitter signal. It makes it last longer. Take someone who's clinically depressed, and the drug of choice these days is to give them an SSRI. So you look at that. Ooh, here you've got the psychiatric disorder. Someone is clinically depressed. You throw in a drug which makes serotonin last in the synapse longer and send more messages, and the person gets better. What your hypothesis has to be, there's too little serotonin in the first case in depression. The serotonin hypothesis, which is a dominant one. Okay, so you give somebody an SSRI, and they're about as effective as the drugs are out there, which means it is by no means guaranteed it's going to help you. Nonetheless, they're very effective. But as always, there's no free lunch. It comes with a bunch of side effects. Not everybody, but a significant subset of people. And a significant subset, in many ways, the leading side effect from taking SSRIs, hefty doses for a long period, is sexual dysfunction. Okay, so that's a trade-off. So you don't orgasm, but you don't weep silently every time you're alone in a bathroom stall. Take your pick. But what you've got there is this trade-off built around the fact that this is a side effect of SSRIs. Immediately, this raises a problem. What is one of the prime symptoms of depression? Anhedonia, the inability to feel pleasure, the loss of interest in pleasurable things, including sex. Loss of sexual motivation is a classic feature of depression. And now you take an SSRI so that you're less depressed, and a side effect is sexual dysfunction. So first off, like, you can't win either way. Nonetheless, the trade-off is still worth it. But how do you tell? Is it the same type of sexual dysfunction? And the answer is no. In a very, very broad kind of way, the sexual dysfunction you get with depression is loss of anticipation, loss of motivation, loss of, jargon in the business, proceptive arousal behavior. In contrast, what the SSRIs are mostly doing is messing with the plumbing. The arousal can still be there, but all sorts of stuff doesn't work as well. There's a little bit of a decrease in arousal, some loss of libido, but what you mostly get is the dread state of genital anesthesia. There's less sensation there. It's harder to get an erection. It's harder to orgasm, or not at all, or it's a very weak one. Or then you can really be as male who's specializing in problems here that not only do you have problems getting an erection, but once you have one, you are priapic, a great D.H. Lawrence term. The erection, you don't ejaculate, and your erection just sticks around forever, which is much less fun than one might think a priori. So you get all sorts of dysfunctional stuff here going on, not in everyone, not to an intolerable extent, but it's a significant side effect in a subset of people. So we've now got what's going on with this? How do people think taking an SSRI, increasing serotonin signaling in the brain, is causing genital anesthesia and stuff like that? And there's a whole bunch of models for it. The first one is, well, obviously it's got something to do with serotonin. Here's where things get messy. Because it turns out SSRI, selective serotonin reuptake inhibitors of all the different types of neurotransmitters, all of which go through this reuptake process. This drug is selective. It only does for serotonin. Major disappointment in the field. Turned out it wasn't that selective. It also inhibits the reuptake of a neurotransmitter called norepinephrine, another one called dopamine. It depends on which particular SSRI. Paxil is the one that tends to cause the most problems with this. Okay, so maybe the sexual dysfunction side effects have something to do with serotonin. Makes sense. Actually, maybe it's got something to do with norepinephrine or dopamine. Maybe it's got something to do with the fact that SSRIs can disrupt the synthesis of what are called steroid hormones. Estrogen, progesterone, testosterone, including in the brain. So that adds some complexity to it. You've got this thing called the sympathetic and parasympathetic nervous system, which has to be really carefully orchestrated to get an orgasm, first arousal, eventual and the SSRIs almost certainly throw off the coordination, the choreography between these two branches of this autonomic nervous system. You get one additional piece of it. And for this, we need to finally have a discussion here about erections. So, erections in the animal kingdom come in two flavors. Either you're a species who gets an erection by increasing blood flow and something sticks up, that would be a hemodynamic, a vascular erection, or you're a species in which you've got a little penile bone in there and a muscle, an erector muscle, pulls the thing up like you're putting a sail up when you're windsailing, and you got it there. So species are different types. If you got the muscular erection, it happens faster, lasts less time. Vascular, slower, but lasts longer. That's the kind we have. Take your pick. So you've got to have, with these vascular erections, the way you get that is by increasing blood flow to the penis. And the way you do that is you have to loosen the blood vessels there. You dilate them. And the way that is done is with a really weirdo neurochemical messenger called nitric oxide. Nitric oxide causes blood vessels in the penis to vasodilate, and thus blood comes in, and you get that. Same exact thing with the clitoris, where with arousal, you get clitoral engorgement secondary to nitric oxide doing its thing. So both penile and clitoral engorgement involves the vascular system. Ever since I was a young child, I hoped someday I would get to say that sentence. Okay, so you've got nitric oxide in both males and females leading to engorgement, and that is part of it. And what do SSRIs do? They inhibit the synthesis of nitric oxide. They inhibit an enzyme, shockingly, bizarrely, an enzyme called nitric oxide synthase, and you inhibit it with your SSRIs. You make less of the nitric oxide, and you don't get your engorgement, your protuberance of your penis or clitoris and all, and that's part of the problem. Okay, so that's what we got here. So that's a side effect. But what the questioner brings up is something else, which is you get over your depression, which most people do when the SSRIs do their thing, and it's great. And you can taper off and eventually you stop your meds, but the sexual dysfunction is still there. And this introduces some great jargon. We have an iatrogenic disease, a disease caused by a medical intervention. You fix something somewhere, and here's the problem. And this is PSSD, Post SSRI Sexual Dysfunction. Notice we have the incredibly rare case PSSD, an acronym for Post SSRI, an acronym within an acronym. It doesn't get better than that. So PSSD, lots of people have the SSRI mediated sexual dysfunction that lasts for a long time afterward. It's just getting recognized to be a problem. What could be going on? The most hand-waving, useless explanation out there is, ooh, it involves an epigenetic change. That's a very flashy, jargony term. And all it means is something has changed in your gene regulation that's now going to last long after you stop taking the SSRIs. A totally bizarre, ironic outcome of you started with a depression, too little serotonin, and you took these drugs that boost up serotonin signaling, and now in the aftermath, you've got too much serotonin. Whoa, be careful what you wish for. That assumes, first off, that's what's going on in these folks. That assumes that's the cause of it rather than norepinephrine. But there's some suggestion now of ironically going in and trying to tamp down the serotonin signaling or throw in some extra nitric oxide or whatever. But what this mostly shows at this point is unexpected consequences, trade-offs with everything. This is a long-term trade-off in a subset of people who take SSRIs where nobody currently knows what it's caused by or how to fix it.
Okay, so now we're going to talk a little about a classic Robert Sapolsky topic, which is stress. So we have two connected questions here. Claudia from Romania asks, is there such a thing as a healthy dose of stress? Can you habituate to stress, or does every stress response cause damage to the brain and the amygdala to grow? Is there any truth to the saying, what doesn't kill you makes you stronger? And then JP from Minneapolis says, I've always heard that stress suppresses the immune system because cortisol is anti-inflammatory, but you've said in some talks that acute stress may actually temporarily boost immune health. Can you elaborate on the relationship between stress and immunity and explain mechanistically how chronic stress can have the opposite impact of short-term stress in some areas?
Two great questions that are highly interconnected. By the way, the name cortisol, that's the name of the main glucocorticoid stress hormone in humans. So for our simplifying purposes, secreting cortisol is synonymous with what your body does when you're stressed. Okay, so everybody knows by now we're about 90 years into stress being a medical term. Stress is bad for you. Stress is bad for you. Every outpost in your body, chronic stress can make you sick, make you more vulnerable to other sources of disease, make it harder to repair. All of that stress, bad news, stress, bad news. The question is stress always bad news? And the answer is no. And when you think about it, it actually makes intuitive sense. So you take somebody who's now dealing with the notion of stress. What is stress? Stress is an environmental, a physical, a psychological challenge to your equilibrium. And your body then has to do some stuff, including secreting cortisol, to reestablish equilibrium. Jargon from ninth grade biology, to reestablish homeostasis. Jargon from biology in the last 20 years or so, to reestablish allostasis. Okay, so a challenge comes along. So are all challenges terrible? Are all challenges bad news for you? And absolutely not. And what we have here is the classic demonstration of this for people in the business of what is called the inverse U relationship. We've got increasing amounts of stress, increasing amounts of stimulation, increasing amounts of cortisol, increasing amounts of anything. And you see something that makes total obvious sense. If you got too little of it, you're bored out of your mind. If you got too much of it, you're stressed out of your mind. And if it's the optimal in between, it's great. We love it. We call it stimulation. What is stimulation? That's the correct amount of stress. It's a challenge. It's a challenge that's challenging enough so that you're not bored, but it's not so challenging that you develop a sense of helplessness. And whether you are a developmental psychologist or a gerontologist and everything in between, you know that too little stimulation is just as bad as too much. Okay, so we've got that. And we wind up thus having in the middle is this concept of good stress. Good stress, what constitutes a good stressor? Some of its parameters. It's not too severe. It doesn't last for too long. Roller coaster rides are not 3 weeks long, that kind of thing. They occur with an overall setting of benevolence. You get on the roller coaster, there's a good chance you're going to feel queasy. It's not likely you're going to be decapitated. It's an overall safe environment. And under those conditions, what are we doing when we're stimulated? We're willing to give up a little control and a little predictability to be surprised. Massive amount, not so good. None whatsoever, not so good. And that's in a gradient with all sorts of stuff. I mean, you take a baby and you never touch the baby, and that counts as the wrong side of this inverse U. And you like scratch the baby's belly now and then, it's that stimulation. And now you scratch the baby's belly with sandpaper. And most people would say that's probably on the extreme side. It's the same just differing intensities, too little, optimal, too much. So we now have this issue of a lot of stuff in the world of stress has this inverse U relationship. And some of the time, what we're most aware of is the difference between the right amount and too little. Some of the time, what we mostly focus on is the right amount and too much. So what could be some of these circumstances where we see it comes out with the stress system is how severe the challenge is to your equilibrium, how severe the stressor is, and how long it goes on. And one of the themes that you see is short term in the early phases of a moderate stressor. Most of the stuff that goes on then is good. You increase the amount of glucose and oxygen delivery to your brain. Your synapses become more excitable. You release more of this neurotransmitter dopamine, which is about anticipation and motivation and reward. Your memory is enhanced. You get just the right level of sensory vigilance. That's you're being stimulated. You're stimulated and things are working well. Your brain's being washed in good stuff and you feel motivated and your memory is good. And what we see with all of those is if it goes on for too long, every one of those benefits not only goes back to baseline, but winds up going below. Chronic stress, and there's less glucose and oxygen delivery to like some very vulnerable parts of your brain. Chronic stress, you get depleted of dopamine. Why chronic stress can lead to depression. Chronic stress, you get memory problems. You get vigilance that is so vigilant 24/7. You get an anxiety disorder. Okay, so we've got our optimal curve. So we see that in terms of, is all stress bad? No, you want to get just the right amount. And we love it. We pay money to have the optimal level of stress to be stimulated. And this exactly applies as well to the inflammatory system. And what you see is massive amounts of stress hormones suppress inflammation, moderate amounts enhance it. The same exact picture there. Okay, so we see these different patterns here. So the question becomes, as raised in the second question, what are the mechanisms? What's the nuts and bolts to explain what's a very tough problem? We're accustomed to the idea, the more of whatever, the more of whatever you get. More of the stimulus, you secrete more of this, or you secrete less. There's a linear relationship, and instead, there's this weird inverse. There's an optimum right in the middle there. How does the brain pull this off? And there's two totally cool mechanisms for this. First off, so we've already heard that you secrete a hormone like cortisol, and it gets into your cells, like your neurons in the brain, where it binds to a receptor for cortisol. And the way it works is that this receptor in cortisol goes and does something to your genes. Critically, in parts of the brain, there are two different types of receptors for cortisol. One of them is a lot stickier for cortisol. Jargon, it has a much higher affinity. It has an order of magnitude higher affinity. It is much better at grabbing onto any cortisol coming in through the bloodstream. So what we got here is this is receptor type number one. And as you begin to raise levels of cortisol and stress and stimulation, it rapidly fills up. And soon it's 100% full and it's maxed out. And what does this one mediate? All the good effects, all the great short-term moderate stuff. And it's only when you've now been churning along for minutes, hours, decades, or whatever, that you begin to secrete enough of it so that the much sloppier type 2 receptor begins to bind them. And what it does is it offsets all the good stuff that type 1 is causing and more than offsets it. And it's this 2 receptor system, which is so beautiful and elegant. And that's a means by which, the more of this, the more you get a straight linear system. Have 2 messengers with very different sensitivities, and you can get all sorts of interesting curves and sigmoidal stuff like that, which is totally elegant stuff. And what that raises is all sorts of interesting possibilities. My lab years ago, for example, did some gene therapy so that in the brain, we boosted up the amount of number 1 type receptor in certain brain regions, and we made rats who had better memory during stress, things of that sort. Okay, so that's the first one. Here's how you differentiate between how long has the stressor been going on. It's only when it's chronic that you start filling these up, or how severe is the stressor, you flood the system. There's one additional aspect to this that winds up being relevant to the inflammation type thing. So short-term stress does a different thing to inflammation than long-term stress. But what you also see is it also depends on which part of the body or which part of the brain. Turns out even a moderate amount of stress and cortisol secretion increases inflammation in 2 parts of the brain. And these are pretty cool parts, the hippocampus and the prefrontal cortex. And meanwhile, it doesn't do that at all in other areas like the hypothalamus. Whoa, how does that happen? It's the same hormone getting to all 3 areas where it has the same type of receptor, the same either type 1 or type 2. It's got the same, it's the same receptor. And in this part of the brain, it increases inflammation, but the same exact thing in another part doesn't do it. Or you could get an even more dramatic example. If you're under lots and lots of stress, a part of your brain called the amygdala pumps out a growth factor, which causes the amygdala to get bigger and more excitable. And that's how stress gives you an anxiety disorder. And that's due to the release of something called BDNF, brain-derived neurotrophic. Meanwhile, next door in the hippocampus, lots of stress, and the hippocampus atrophies and memory gets worse. And that's because you decrease the levels of this brain-derived neurotrophic factor. How do you do that? It's the same hormone getting to the amygdala and the hippocampus, where it's got the same types of receptors, and they do absolutely opposite things. And here's where you see one additional cool piece of the story. What you have is, okay, here we've got in the hippocampus, the cortisol in both the hippocampus and amygdala comes in and binds to the receptor, where the hormone receptor complex goes to the DNA and has an effect on this gene. And what we see is in the hippocampus, it inhibits this gene from that growth factor. In the amygdala, it does exactly that. How does it do that? Because of these things called receptor cofactors. Floating around inside the neurons in the amygdala is the green one, and inside the hippocampus are the orange ones. When the green one latches on, it throws the on switch here. When it's the orange one, it throws the off switch. So this begins to give you the means of, well, what does stress do? It depends in what part of the body, how severe of a stressor, and how long it goes on. This is totally cool mechanistic stuff, how you can pull off like opposing effects and things of that sort with inverse U's. What it mostly is telling us, though, is something halfway between is where most of us settle out. Now, what that raises, of course, is the issue of everybody doesn't have the same inverted U shape. Some people, their optimal stimulation point is just a little bit of challenge, and other people, it takes a mass individual differences. One person's idea of like a reckless, like really push the envelope day is to go bird watching in Central Park, and somebody else is to like sign up to be a mercenary in Yemen or something. Individual different shapes to the where's the optimal point. Where's that coming from? Genes, fetal environment, early upbringing, all that sort of stuff. What particular shape do you have to that curve? Which brings us to the final question there. Well, if some stress turns out to be good for you, is there some precedent for that whole notion of what doesn't kill you make you stronger? And this is a version of what that would look like. Here we have a moderate. Is that moderate? Here we have a manageable stressor. And what you see is you're functioning here. And afterward, in this Kelly Clarkson scenario, afterward, what doesn't kill you make you stronger? That's wonderful. That's all of that. That's not what you necessarily see. And what the rule is, it all depends on where you start off. If you start off downtrodden, what doesn't kill you makes you stronger isn't what occurs. Whatever happens pushes you down further. What doesn't kill you makes you stronger is a luxury of the lucky. If you're already starting off in good shape, adversity is an opportunity for growth and resilience and all of that. For most people out there, all you're doing is being kicked further down. So that isn't necessarily the case all the time.
And that concludes episode 30. So keep submitting your questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Share Sapolsky, and thanks for your continued support of Science and the Beard.